Hello everyone, welcome back to the Vital Education Group. So, as you all know, the channel name has been changed from Vital Biology to the Vital Education channel. So, there is no any confusions that we will be taking all the classes similarly, but there is only a small change, that is channel name. Okay, there will also be biology subject also. Okay, so there is no any confusions in that. So before 2 weeks back I have stopped until the megasporogenesis, right? That is in chapter 2, sexual reproduction in the flowering plants. So I'll just continue the chapter uh, where I stopped until the megasporogenesis, right? So today let us start with the subunit of chapter 2, that is pollination. Okay, so first let us see that the definition of the pollination. So the transfer of the pollen grains from the anther to the stigma of the pistil is called as pollination. So as you know, I have already mentioned you and I have also showed you that what is pistil, pollen grain, anther, stigma, everything, right? So this stigma and pistil are the female reproductive parts of the flower, right? And so 1 second, I'll just again show you that. Okay, so can you observe here? So this pinkish like structure, that is pistil. Okay, that is female reproductive part, and this bluish structure here and all, you can observe this sky blue color structure, that is male reproductive part in the flower. So this is called as stamen. Okay, so which consists of anther and style filament. Okay, so and this pink color is female reproductive part, that is pistil. Okay, so here this transfer, as I told you, the pollination is nothing but the transfer of the pollen grains from the anther to the stigma of the pistil. So right, that is right. So here you can observe that from the anther, these yellow colored structures, spherical like structures, that is called as pollen grains, that are transferring from here to this female reproductive part, that is pinkish part. Okay, here you can observe that pistil. Okay, so here this yellow color part or like pollen grains are shedded. So here also you can observe. So I guess now you have remembered that what and all I have teached about this pollen grains, anther and all, right? So let us go back to the topic. So as I told you, pollination is defined as that the transfer of the pollen grains from anther to the stigma of the pistil is called as pollination. So next let us go to the kinds of pollination, means types of pollination. So mainly there are 3 types of pollination. There are other types of pollinations too, but let's take only the main pollinations, that is 3 types of pollination. So first one is that autogamy. Okay, and the second kind of pollination is coitonogamy, and third xenogamy. Okay, so 3 main kinds of pollination are autogamy, geotonogamy and xenogamy. So first let us see that what is autogamy and its examples. Okay, so auto, nothing but own. Okay, self. Gamy, pollination, transfer. Okay, so here the transfer of the pollen grain from anther to stigma, that is common. Okay, pollination means this is common, transfer of pollen grains from anther to stigma, transfer of pollen grains from anther to stigma. But the main difference is that of the same flower, in the same flower itself, the transfer of pollen grains from anther to stigma, it is transferring in the only one flower, that you can observe here. So can you see this flower? Okay, so this is self pollination, nothing but it's also called as autogamy. Okay, autogamy is also called as self pollination. So here you can observe that in a single plant, in the single flower itself, you can observe that these pollen grains are transferring to the stigma of the pistil of the same flower. In the same flower itself, the pollination is taking place. Okay, so this is self pollination or else autogamy. And example for this self pollination is that viola and oxalis. Okay, viola and oxalis. Okay, so let's move on to the next kind of pollination, that is gyautonogamy. So what is gyautonogamy? The transfer of the pollen grains from anther to the stigma of the another flower of the same plant. So here the flowers are different but the plant is same. Means in the same plant but in two flowers there is a pollination taking place. So I'll show you that picture also. So can you observe here? So here is one of the plant. So in this plant, so in this green line plant, so there are four flowers. So in that, let us take these two flowers. So can you observe here? Here also there is anther and stigma, and in this flower is also there is anthers and stigma. So here these pollen grains that are transferring from here to another flower, but the plant is same. Okay, the plant is same but flowers are different. Okay, so this is called as gyautonogamy pollination. Okay, gyautonogamous pollination or else gyautonogamy pollination. Okay, so same plant but different flowers in the same plant. Okay, so now let us see the once again let us see the definition of the gyautonogamy. So here the transfer of the pollen grains from the anther to the stigma of the anther flower of the same plant, okay, but different flowers. So example for that is cucurbits, okay, commonly called as cucumber plant. Okay, so next let us go to the third kind of pollination, that is xenogamy. So here what type of pollination takes place? So the transfer of the pollen grains from the anther to the stigma of another flower of the different plant. Okay, so here the plant is also different and the flower is also different. So can you observe here this green colored line, that is one plant, okay, and here this is another plant. Okay, can you observe here? So here there are two plants. So here in this plant let us take one of the flower, and in another plant one of the plant let us take for the xenogamy. Okay, so here this flower, that is here also. So here the pollen grains are transferring from one plant to the another plant flower. Okay, so here the plant is not same and the flower is also not same. The flower is also different and plant is also different. So this type of pollination is called as cross pollination or else xenogamy. Okay, I'll just explain it once again. The transfer of the pollen grains from anther to the stigma of another flower of the different plant. Okay, just understand this very easy topic. Okay, an example for this xenogamy is that papaya. Okay, so this much is about some pollination. So next, as I showed you these diagrams, again I'll be explaining now. So this is self-pollination, also called as autogamy. As I told you, the transfer of the pollen grains from anther, okay, here from anther to the stigma of the same flower. In the same flower itself this pollination is taking place, just here itself. So this is called as self pollination, and for self pollination there is no any requirement of any insects, wind or anything for the population purpose. It just independently pollinates itself. Okay, there is no need of any agents. Okay, and the next is gyautonogamy pollination. Okay, so here as I told you, the transfer of the pollen grains from the anther to the stigma of the different flowers but in the same plant, but different plants, the transfer of pollen grains is taking place. Okay, this is gyautonogamy. Okay, next cross pollination, it's also called as xenogamy. Okay, here in cross pollination, as I told you, the plants are also different and flowers are also different. So here the transfer of the pollen grains from the anther to the stigma of the another flower of different plant. So here is one plant and this is one of the plant. So this is a simple explanation about this thing. So next let us move on to the next slide, that is about the agents of pollination. So agents is nothing but the what and all the things that helps during the pollination, during the transfer of pollen grains. Okay, so let us see that what and all helps for the pollination. So first let us see that abiotic factor. Abiotic, nothing but non-living factors, non-living agents. Okay, biotic means living agents. Okay, so first let us see non-living agents, that is abiotic agents, that is with the help of wind the pollination takes place. So if the pollination takes place with the help of wind, then it is called as anemophily. Okay, then it is called as anemophily. And next, if the pollination takes place with the help of water, then it is called as hydrophily. Okay, these are the two abiotic agents of pollination. So next let us see biotic agents of pollination, living agents of pollination, that is insect, birds, bats, mammals, and some snails also we can take. Okay, many are there. So if the pollination takes place with the help of insect, then it is called as entomophily. Listen it carefully, it's so easy if you listen it carefully. Okay, so biotic agents there are many factors, that is insects, birds, bats, and like snails, mammals, everything. So if the pollination means transfer of pollen grains takes place with the help of insects, means then it is called as entomophily. Okay, so like a study of insects is called as entomology, right? So here if the transfer of pollen grains means if the pollination takes place with the help of insect, then it is called as entomophily. And if the pollination takes with the help of birds, then it is called as ornithophily. And the pollen grains which are transferred with the help of bats, then it is called as chiropterophily. Okay, chiropterophily. And next, with the pollination that if the pollination takes place with the help of mammals, it may be human beings too. Okay, then it is called as zoophily. Okay, this is a important slide, you just want to keep that in mind, it's a very easy thing. So next let us move on to the next slide. So you can observe here, so these are the abiotic factors, these two, that as I told you, wind and water. Okay, and this side that you can observe that biotic factors. Okay, non-living factors and living factors. Biotic means living. So as I told you, wind. Okay, wind pollination, water pollination. Okay, so in wind pollination, so there if any flowers that pollinates with the help of wind, then that pollen grains and anther that are loosely attached to the plant, flower. Okay, so let's move on to the next slide, that is adaptations in the flowers for pollination. So how and all these flowers are adapted to the pollination with the help of agents, let us see. First let us say take wind pollination. So in wind pollination, the flowers pollen grains that are present in the flowers that are light, loosely attached, and non-sticky, winged. Okay, so these are non-sticky and winged, means like wing like structures will be present attached to the pollen grain, so that in when this wind comes fastly, so that they can fly and shuttle down somewhere else, shed down somewhere else. Okay, so that is wind pollination. And these pollen grains in the wind pollination are very light, means weightless, and non-sticky and wingless or winged. Okay, and here in this wind pollination flowers, the anther is well exposed. Okay, so this anther is well exposed means it is very loose and very loosely attached, as I told you. And next, a stigma, it is very large and feathery. Okay, And example for this wind pollination is corn cob and grasses. Okay, so next let us move on to the water pollination. So in water pollination, these pollen grains are very long and ribbon like structures. Okay, very long and ribbon like structures. Okay, these pollen grains are okay, and these pollen grains are protected by the mucilaginous covering, means like jelly like structure that covers the pollen grains in water pollination. And next point, that is it produces the large number of pollen grains. So in water pollination, the pollen grains are very large when compared to wind pollination. Okay, so next let us move on to the insect pollination. So in insect pollination, the flowers are very large and colorful and fragrant, nothing but it's so smelly, and the odor is very good and rich in nectar, means like honey. Okay, so that is rich. And the pollen grains and stigma are sticky because the insects that sticks rubs on that pollen grain, and it just sticks that pollen grains to its body, and it just transfers to when it sucks the nectar to from another flower. By that time, the sticked pollen grains that falls on the another flower. Okay, so I'll just show you that insects once. Okay. So let us take honeybee as one of the insect. Okay, so honeybee is one of the insect, right? So here you can observe that it this yellow color, it has just stuck to the insect body, to the honeybee body, right? So this will go and sit on the another flower for the nectar purpose. By the time this pollen grains falls on the flower another flower's stigma. Okay, so let us move back to the main topic. So this is about the insect pollination. So next let us move on to the next slide, that is outbreeding devices. So let us see what are the mechanisms and devices that promotes the cross pollination. Okay, so actually self pollination is not much better when compared to cross pollination. So cross pollination is very good compared to self pollination, means autogamy or else guatonogamy. Okay, when compared to autogamy and guatonogamy, the xenogamy is best. Okay, and it is a hybrid and can produce a variety of different flowers. Okay, so here in outbreeding devices, plants have many mechanisms and devices too that promotes the cross pollination. For the help of cross pollination, means for the xenogamy, there are many devices and mechanisms. Okay, so let us see what are those mechanisms and devices. First one is dichogamy. So in this dichogamy mechanism, the stigma and the anther mature at the different time, so that there will be cross pollination. Okay, so this is one of the type of device. And the another one is heterostyly. So in heterostyly, the stigma and the anther are placed at the different levels or else different environmental conditions, so that also they can mature in a late stages, in the different stages. Okay, so next device, that is self sterility or else self incompatibility. So this is a genetic mechanism that prevents the self pollen from fertilizing the ovule by inhibiting the pollen germination. So this self sterility that prevents the self pollination, means autogamy. As I showed you what is that autogamy means, as I told you, the transfer of the pollen grains from the anther to the stigma of the pistil of the same flower. So that should be avoided, because the variety of flowers will not be produced if there is a self pollination. It should be some cross pollination from different plants and different flowers. Okay, so let's move on to the fourth device, that is unisexuality. Okay, that is dioecium. The plant produces either male or else female flower. Okay, so this is another device. So let's move on to the next slide, that is artificial hybridization techniques. So there are two artificial hybridization techniques that are given in your textbook, but there are more techniques that are used for the artificial hybridization. Okay, but here they have mentioned in second PU biology book, they have mentioned only two artificial hybridization techniques. So first one is emasculation technique and the second one is bagging. These two are the artificial hybridization techniques. So let us see what is emasculation and what is bagging. So emasculation is nothing but the removal of the anther from the bisexual flower before the anther matures. That is called as emasculation. So the removal of the anther, okay, before it matures. If there is if they remove the anther before that anther matures in the bisexual flower, then it is called as emasculation. Okay, so next let us move to the next one, that is bagging. So the emasculated flower that is then bagged or else covered with the bag to prevent the unwanted pollination. If there is somewhat unmatched pollen grains that shed on stigma, then it will not be any pollination germination cannot be takes place. So this should be prevented, right? So for this type of prevention means unwanted pollen grains that should not be seated on unknown stigma, right? So for this prevention or else to prevent this type of unwanted things, so the bagging technique is used where the cover is closed to that flower so that the pollen grains cannot fall into that stigma. So this is the process called as bagging. Okay, so next, pollen pistil interaction. So what is pollen pistil interaction? If you know that what is pollen grain and what is pistil, so then it's very easy to understand that what is pollen pistil interaction. So actually I have already told you that what is pistil, what is pollen, right? So you can observe here this red colored roundish structures, so that is pollen grains, and this elaborated structure, that is pistil. Okay, female reproductive part in the flowers, so that is pistil. So first let us see that definition for the pollen pistil interaction. All events from the pollen depending on the stigma until the pollen tube enters the ovule are together called as pollen pistil interaction. Did you understood? So if the pollen deposition means these pollen grains come and deposit on the stigma until this pollen grains germinate and it just interacts with the, so can you observe here, these are germinated means the pipe like germ tubes are formed, pollen tubes are formed, and these tubes are germinated and they are interacting with the ovule, right? So this together means this together like pollen tubes and this ovule together, these are called as pollen pistil interaction. Interaction is nothing but communication like purpose. Okay, so next, so how this process takes place means pollen pistil interaction process, how it takes place. First it recognizes that is the pollen that is suitable for me, means like for stigma it is suitable or it is unknown. So yes, it is suitable means it just gives a permission to germination of the pollen grain, or else it is, if it is somewhat unwanted pollen grains means it just blocks the hormones like which should not germinate, where the pollen grain should not germinate, it just leaves as a stable. Okay, so here the process is first it recognizes, okay, whether it this pollen is matched suitable or not, and then germination, it gives the permission for the pollen grain to germinate and pollen tube growth. So here this pollen grains have germinated and release the pollen tubes into the ovule. So this is a pollen pistil interaction. Okay, so this much is today's class. So in the next class I'll be continuing with the double fertilization. Okay, so thank you so much, and for the more updates just subscribe my channel and press on the bell button for the more updates. And if you need any I'll be providing the notes and some PDF materials and something like any usable study materials in the Telegram channel, that is Vital Education Group, that I'll be providing below in this video's description box, so that you can join me there by using this link in the description box, and there you can communicate with me. Okay, so thank you so much. Bye bye.